Okay, so welcome to my GIT Physiology lecture. I would like to introduce myself first. I'm in the 5th year and I volunteered to do this one because I do genuinely enjoy the GIT Physiology. And also the physiology department probably likes it. They keep calling the GIT the mini brain, if you have noticed. And we'll get to that later, why they call it so. So the outline would be like this. I would love to cover everything, but I don't think I can. But I would try to include that what's important. So I would start from the up to the bottom. So from like saliva, also swallowing, stomach, and then stomach, duodenum, and also the small intestine, the colon, and also defecating as well. And what I missed here was the absorption, which includes like water transport, peptide transport, that kind of thing. And I thought that you could study that on your own by doing biochem, because I guess the biochem lectures or like the textbooks are better at that. And I would like to concentrate on the physiology here today. Okay, wait, I would like to see how many people are watching now. Okay, 6. Okay, so I would like to start. So what is the GIT? So we would probably know that it's from the mouth to the anus, but we would have to include the accessory organs, which include the glands, the salivary glands, and also the pancreas, which secretes important peptides, enzymes for digesting, and also the bile. And bile is secreted by the liver. So liver is also including in the GIT system. And GIT, what the job is to digest and to absorb and to excrete anything you don't excrete the waste. And for these three to coordinate properly, we have a lot of good system that we have in our body, and we'll get to that later. So we have a lot of reflexes that it does on its own. So those are the local reflexes. We also have hormones that are secreted by the GIT, and also we have our brain that we can control it time to time. For example, defecation and swallowing and so on. So first of all, I would like to go on to the saliva. Saliva. So saliva is probably, you learnt it from anatomy, that it's secreted by the salivary glands in our mouth. And we have the major salivary glands and the minor salivary glands. And then the major ones, we have the parotid, submandibular, and also the sublingual. And for the minor ones, we have a lot of them. But what are the big differences are, the minor ones are always secreted. No matter what you're doing, it's always secreting. Whereas the major ones are the ones that are responsible for creating saliva when it's needed. So when you're eating, you have to secrete a lot. When you're thinking about food, that's when the major salivary glands are working. Yeah. And its function is not only to make the food more chewable, it also has a lot of antibacterial things that protect us against the bacteria in our mouth. And also it has an enzyme called amylase, which digests the starch or carbs already in the mouth. And we would have to know how it's formed. So we have the acinar cells like this, and it's secreted obviously from the blood. And first of all, it is, well, I would like to say at the end, it will be hypotonic. That's what's important. And what does it mean? It means that there's water here, and they get rid of the Na, sodium, and chloride. Yeah. and that makes it hypotonic. And since the water is impermeable here, it will remain hypotonic. And instead, you will get this hydrogen carbonate, and this makes the saliva slightly alkaline. And why are these two important as the characteristics of the saliva? So it's important that it's hypotonic so that you can taste. If it's hypotonic, if it's salty, you won't be able to taste food properly. And alkaline, why? Because since the stomach produces a lot of acid, there is a lot of chance that it does go up to the esophagus. So we want to neutralize it as much in the esophagus. So it is alkaline. So it's better to remember that it's hypotonic and alkaline. And this is physiology, not anatomy or biochem. So we have to know how the saliva formation is regulated. So when we're secreting a lot of saliva, first of all, the blood circulation, the blood supply is very much increased, and that's why you get to secrete a lot of water, secrete a lot of saliva. And there's a specific, you have to know, wait, let me look for my notes. So you have to know the trigger of secretion and also the what inhibits the saliva formation. So you see, if you think about daily life, when do you get a lot of saliva? So first of all, there's this phase that you haven't even eaten food, but you would smell it, taste it, you would smell it, hear it, however you like to imagine. And by doing that, it stimulates the high centers of the brain, and it goes to the salivary nucleus in the medulla in the brain, the medulla oblongata. And this stimulates the parasympathetic nerve to different ganglions depending on which gland you want to stimulate. So, for example, if you want to stimulate the parotid gland, that parasympathetic would stimulate the otic ganglion. And by releasing this ganglion, releasing acetylcholine, it stimulates the parotid gland. And in the same way, it activates the submandibular ganglia to the submandibular gland. So that is the way. And also you should know what inhibits this salivary formation, salivary secretion. It is when you're sleeping, you have less. When you're in fear or when you have an exam, when you're in a fight, that's obviously where you're in a sympathetic mode when you're not eating. That's when it's inhibited. So yeah. So I think I should say it again. So the salivary gland regulation, secretion regulation. So it is by the higher cortex from whatever you're feeling, whatever you're seeing, and it's either positive or negative. And it stimulates the salivary nucleus and in the medulla oblongata. A lot of nucleus are in the medulla oblongata, these very essential things that you do in everyday life. And by that, it stimulates the parasympathetic, obviously, because you want more of the saliva. And depending on which gland you go, so either parotid, submandibular, you go to the different ganglion and release ACH, acetylcholine. And that causes vasodilation of the gland, and you will get more secretion. Yeah. So I think what's important in this question is that you know the regulation and also the hypotonic and alkaline characteristic of the saliva. Okay. Next. So I would like to talk about the GIT, tract structure in general. So we're talking about the stomach, the small intestine, and then the large intestine. And the basics are all the same. So if you look at the lumen. You have this lumen, and then you have this simple columnar epithelial cells, and underneath that, that's submucosa. So you would have a lot of glands there. And yeah, this is the glands that you could see. And then near the gland is where you have the submucosal plexus. Yeah, submucosal plexus. And you would have 2 layers or 3 layers of muscle outside of that. So either way you would have the circular muscle and the longitudinal muscle. So this and this, yeah. And between that we have this mesenteric plexus. So 2 things you have to remember. So we have the submucosal plexus and mesenteric plexus. This is the innervation that they get. And yeah, and to talk more about the submucosa plexus, so this, in the name it says submucosa, so it is in the submucosa. And what else is in the submucosa? The glands. So it means that it stimulates the glands to secrete. So in like physiology, they say like submucosal plexus is responsible for the secretion of the glands, whereas the mesenteric plexus, which is in between the circular and the longitudinal muscle, is responsible for motility, which is like peristalsis and stuff. The movement of the gut is by this mesenteric plexus. Yeah, and in the stomach we have the oblique muscles, but it's pretty much the same, the mechanism. And in the stomach we have this oblique ones because not only that you want to carry it, you want to mix the things inside the stomach, which is a big bag, so you gotta mix it properly. So you have the oblique for that, is how I remembered. Probably I'm saying it not the right way. And what else? So and then the outside is, as we know from histology, either it's called the serosa or adventitia, depending on what's outside of it. Okay, I hope I didn't miss anything. Yeah, so I also wrote it here, the most important thing. They love asking you this. Yeah. Okay, so next slide. Oh, when I was studying physiology, I used to get very confused with this. So there's different types of phases of digestion, and they love using this word cephalic phase. And cephalic means brain or like the head. And it's the phase when you haven't eaten yet, but you're thinking about food. Your food is in front of you. And when we talk about eating, it's already a phase because a lot of things are happening already. So in more details, what's happening is that your vagus nerve is already stimulated. So you would have more secretions of saliva. And also it stimulates the hydrogen. It's more acidic in the stomach now. And it releases more gastrin. Yeah, we'll get to it later. So gastrin is also responsible for the acidity in the stomach. And then we have this gastric phase. This is probably the phase that everybody would think of when we're eating. So this is when our mesenteric nerves are activated. How it's activated is that when a food gets in, and it's a local stretch receptors that senses that, oh yeah, food's here, and it activates the mesenteric nerves, and then stimulates the local reflex of like peristalsis and so on. Vasovagal reflex. This is any reflex caused by the vagus nerve, and the vagus nerve does pretty much everything for the GIT. And oh yeah, and also what's important is that yes, vagus is really important in the gastrointestinal system, but it only innervates from the up to the upper large intestine. So from the lower large intestine, the pelvic what was it called? not the pelvic nerve, but the pelvic, sorry, I'll get back to it, but the pelvic nerve or the pelvic plexus are responsible for it. Yeah. And then when you're eating, you have these hormones secreted. We'll talk about it later. And this is the intestinal phase where your gastric content goes to the intestine. And now what you have to do here in the intestinal phase is that since you secreted a lot of acid in the second phase, you have to neutralize it or put it back to normal. Yeah. Or this is the phase when you activate the system from stomach below so that they're gonna be ready for the food that comes. Okay, so yeah, so this slide just wanted to say that they like to say the cephalic phase, gastric phase, intestinal phase, if you have to separate it in phases when you talk about eating. So, okay, so this is the stomach. And we will get into details because it's very important. And so, I don't know if you remember the anatomy, but this is the cardia. And this is where the esophagus has to be. So there will be the lower esophageal sphincter here. And this is a very important sphincter. We'll talk about it in the at the end of the class. So food gets in, and this is the fundus of the stomach. And this is the body, the main part. And this is the body, antrum, and pylorus at the end. And pyloric sphincter is the sphincter that controls whether to leak the content to the duodenum or not. Yeah. So if you have a really fatty meal, this pyloric sphincter is constricted, or the releasing of the content will be really slow. Whereas if you just drink water or something really easy, then it's just gonna go flow. Yeah. And yes, anatomy is important, but what's important is that you have to know which cells are in which part of the body. Yeah. So for example, gastrin, G cells that are responsible for secreting gastrin are found in the antrum. Yeah. And yeah. And also, ghrelin is a hormone for, it's basically a hormone that tells you that you're hungry. And that is also found in the antrum of the stomach. And what happens is that, I don't know if you know this, but there's a surgery called gastric bypass surgery. It's done for obese people, and they're basically very addicted to food. So what they do is they cut part of the stomach, which is this part. And not only that the size of the stomach gets smaller so that they can't eat as much as they used to, they get rid of the cells that secretes ghrelin, which is the hormone that tells you you're hungry. So this surgery works for a lot of people, and those are the reasons behind it. Yeah. Okay. And then next, so back to this histology thing. So we have the glands, and yeah, we have the glands, and as we talked, we have the oblique, circular, longitudinal layers, and we will have the submucosal plexus here and the mesenteric plexus somewhere around here. What's important here is that in each pit we have cells. We're talking about the stomach here. And then if you look carefully, we have the parietal cells at the top, chief cells around in the middle, and enteroendocrine cells at the bottom. Yeah, and we learned it in histology, and I also forget it all the time. But this parietal cell is the one that's responsible for secreting HCl. HCl, remember that. I think that's really important. You can get confused with any other cell secreting anything, but the most important thing is that you know that HCl is secreted by parietal cells. And all the other cells are pretty much affecting this parietal cell. Okay? So that's very, very, very important. Okay, this chief cell is the one that's responsible for secreting pepsinogen. This is the one that becomes pepsin. And this one, pepsin, which is the one that you need for protein digestion. Yeah. And this, and also pepsinogen to pepsin. I should go back. So there's a word called zymogen. What is this? This is the inactivated form of enzymes. And enzymes, when they're released, they're usually inactivated at the beginning because the main reason is that you want to activate it at the right time, at the right spot. So you have to you have to release it in an inactivated form. And so the inactivated form are all called zymogens. And so this pepsinogen is the zymogen of the pepsin. And this change is done by the low pH in the stomach. Yeah. And keep maintaining the low pH in the stomach is very important because not only it kills bacteria, but this pepsinogen requires really low pH to be activated to pepsin. So I think those two things are the ones that you should be able to say in the exams. And then we have this enteroendocrine cells. I used to confuse this with enterochromaffin cells, but what it means is that enteroendocrine cells, any cells that secrete hormones in the GIT tract is basically called enteroendocrine cells. And depending on what they secrete, they have different names. So for example, if they secrete gastrin, they're called S cells. If they secrete secretin, they're called S cells. But somehow they confuse you. Like, for example, somatostatin is somehow released by the D cells. I don't know why they're called D cells. But yeah. And also CCK, cholecystokinin, is secreted by I cells. There must be some reasons behind it, but I don't know. But yeah, it's a little confusing. So this is the enteroendocrine cells. They're just a big group of everything, yeah, of all the different cells that secrete hormones. Yeah. I hope I'm not going too fast. If anybody's taking notes, I would wait. Wait, let me see if anybody's talking. Okay. Okay, I'm going to continue. Okay. I know I'm a little mean that I only put this in the slide, but this slide shows everything. I know you will get confused with which cell secreting which and like what's doing what. But if you remember just this, be able to draw this before the exam, then you probably would have a high chance of doing it well. And I also use this for my exam. So what is this picture? So this picture is a picture of the gastric parietal cell. So it's a parietal cell. And to go back, the parietal cells were this. These are the ones that are responsible for releasing HCl. Yeah. HCl. And and these parietal cells are important, and its release of hydrogen is determined by a lot of factors, a lot of hormones, a lot of stimuli. And this is what it's about. Yeah. And first of all, you have to know how this parietal cells secrete potassium. So it secretes by hydrogen, potassium, ATPase. Yeah. So by releasing potassium, it exchanges with potassium, and this is an ATPase. And it's easy to remember because imagine all the H plus all outside of the parietal cell. Yeah. And you have to secrete it even more, even though there's a lot of H plus outside. So it has to secrete against the concentration gradient. And of course, that requires a lot of energy. So that's why it's ATPase. And then that's why it's this. Yeah. And usually this enzyme, if it's not needed, or if they don't have to secrete a lot of H plus, it would just be in the cell. Yeah. And when they get the stimuli like these plus that you can see in the picture, it gets to this lumen, yeah, and starts doing its job. Okay, so then we should talk about what stimulates this H plus secretion. So, okay, we can go from here. So vagal stimulation. So vagal stimulation just means like when you're in a parasympathetic tone. So when you're relaxing maybe, or anything when you have food in front of you, you get hungry. This vagus nerve releases acetylcholine at the end of the nerve, and this acetylcholine binds to the M3 receptor. And same thing with G cells. This is the one that I said that's responsible for releasing gastrin. It binds to a receptor on the parietal cell called the CCKB receptor. I know it's hard, but yeah, we gotta remember that. And these and also, okay, let's talk about this one too. And enterochromaffin cells in the GIT, it releases histamine, which goes to the H2 receptor. And all this three, so gastrin, histamine, acetylcholine by the vagus, stimulates the hydrogen secretion. Yeah, but this two and this one has a different mechanism. So what I mean is that this two, vagus and G cells, or this gastrin, it does this by increasing the calcium level in the cell. Okay, whereas this one on this side of the one, this one is done by CAMP. It's a second messenger. Both of them are second messengers, but they're different. So they're either controlled by calcium or CAMP. Yeah, and this we'll talk about it later. But somatostatin is the one that does the opposite. So when this is released and binds to the receptor on the parietal cell, they decrease the amount of CAMP. So they decrease the secretion of hydrogen secretion. Okay, minus and plus, it means minus here. Plus is plus here. Okay, I hope that made sense. Oh, and also these G cells, ECL cells, this histamine, are secreted when you have that food going on, and it stimulates the release. So what I mean by this, so which one predominates during the cephalic phase? So when you see the food, you haven't eaten yet. Yeah, so you still don't have the release of gastrin or histamine yet, okay? So, and if you get this question, the answer is the vagal tone, so the parasympathetic. Just by vagus, acetylcholine is the one responsible for the hydrogen here. And, okay, what happens if gastric secretion was chronically reduced? I kind of mentioned in this slide that keeping the low pH is very important for killing the bacteria, any bacteria that we don't want in the stomach. So when they have reduced secretion, they easily get infected. And also what happens is that, so the pepsinogen is not activated to pepsin. So you would have digestion problems, and you don't want that. Yeah. And and also I forgot to mention, so not only the stomach releases hydrogen all the time. If it's very acidic, it's gonna even melt itself, and you don't want that. You wanna protect your own mucosa while secreting the H plus. So what it does is it also secretes hydrogen carbonate and also some mucosa around the mucus or mucosal cells, or like protective factors against the acid to the stomach. And so if you have too much acid, or in other words, your protective factors of the stomach goes down, that's when you get like it's called peptic ulcers. It's an erosion of the stomach mucosa. If you think about it, you can probably imagine just having acid on your skin, it melts, it's gonna burn, and that's what happens to your stomach. and it can even bleed, it can even perforate, and it causes a lot of problems. So I think you were all wondering why there were like red lines here. These are the drugs that would stop or inhibit the secretion of acid. Okay, so if you want to stop this secretion itself from the hydrogen potassium ATPase, you could stop it here by a drug called omeprazole. And same system here. So, it seems like too much to you right now, but you would have to encounter this in every subject, like pathophysiology. We'll do it again in pharmacology. So, yeah, it is important. And it's also important that these two are bicalcium. This one is from CAMP. And by stopping different levels, you can stop the secretion. Yeah. And also, what I wrote here is that there's negative feedback. So when you're eating, your food is in the stomach. A lot of acid secretion. But then once it's gone, you want to get back to normal. Yeah. And also you don't want to make it too acidic. And for you to stop that, there's a negative feedback. So these G cells would I don't know much in details, but probably senses the low pH, and the low pH or the acid is the negative, goes back as a negative feedback and inhibits the release of gastrin, which would inhibit all of these cascade. Yeah. Okay. And we'll talk about these again in the next slides. Yeah. I'll ask the comment. Did everybody understand? If you want me to repeat it, I can say it again. If you could say it in the comments. But okay, I guess I'll just continue. Okay, I'll continue. Yeah. So this one. Okay, so pancreas. This itself deserves its own lecture, but I'll just talk about it briefly because it's not really the GIT. It's just we just want the enzymes that it's involved in digestion. So pancreas are responsible both for exocrine and endocrine. And what I mean by that is exocrine is the juice, the pancreatic juice that is secreted from the pancreas that goes to the pancreatic duct and then the common bile duct and then to the duodenum. And it's responsible for basically all nutrition breakdown. Yeah. And since it wants to neutralize all the acidic thing that comes from the stomach, it's alkaline. And it's alkaline by the hydrogen carbonate. And yeah, I think that's good. And I didn't put the list here, but there's a lot of enzymes. You could have a look at your textbook, but there's a lot of enzymes secreted by this pancreas, and they're responsible for different things like this elastase, carboxypeptidase, phospholipase, these kind of things. So for elastase, these are the ones responsible for degrading elastin, carboxypeptidase, and phospholipase. This is also the one that break down lipid. Yeah. And everything, what I wanted to say is that a lot of enzymes all depend activation on trypsin. Yeah, trypsin. Yeah, so if you have trypsin, then these enzymes will be like, okay, I'm going to activate so that I could do my stuff. Whereas then you would think, okay, then how is trypsin activated? So trypsin is trypsinogen when it's a zymogen. And when it gets to the lumen of the intestine, that's when you should be activated, right? So it's activated by this enteropeptidase, which is all in the lumen. Yeah, in the lumen by enteropeptidase. Then now it's trypsin, and then all the other enzymes get activated. And you would so there's this you will learn it later, but for pancreas, there's this thing called acute pancreatitis. And what happens is that this enzyme gets activated in the pancreas. And you don't want that because it's just going to degrade its own self, the pancreas. And what the cause is this phospholipase, because it degrades lipid, and pancreas is basically, there's a lot of fat around it and all that. So if this lipase gets activated in the pancreas, you get really bad acute pancreatitis, which is life-threatening. So what was important here is that there's a reason why enzymes are released as zymogens, and you should know which is activated by what. But at the end, the beginning starts from trypsinogen being trypsin by the enteropeptidase. Yeah. And I wrote here sphincter of Oddi because this is the sphincter that controls the pancreatic juice to the lumen of the duodenum. And it won't just release it when you haven't eaten for like half a day. And what's the point? It releases when the food is around the stomach, so it gets ready, and also when there's food for that. So sphincter of Oddi relaxes when there's food, so it can digest. Yeah, and vice versa. It's constricted if not. Yeah. Okay, and bile also, I will talk briefly about this. So bile is also important for digestion, especially for fat, and it's done by it's an important thing to do emulsification. Yeah, this word probably from biochem. And what else is important? It's important for excretion of lipid soluble xenobiotics. This is also biochem, but what it means briefly is that when you have any toxin or any waste that you don't, any toxins that you don't desire, it excretes, if it's lipid soluble, it secretes with bile. There's no other way. Or maybe by stool if you drink it or eat it. But yeah, so this is important for excretion of toxins, xenobiotics. Yeah. And also bile has a golden yellow color, and this is because of bilirubin or biliverdin. And what was this? It was the breakdown product of hemoglobin from red blood cells, if you remember. Yeah. So when hemoglobin is broken down, it becomes heme and globin. And since globin is protein, it's going to get recycled. Whereas heme, it will be, become the bilirubin or biliverdin in the liver, and it will be secreted as bile. And then it would still continue its journey in the intestine, can be reabsorbed again and go to the liver, or can be secreted as can be excreted as feces. Yeah. So bile, and also it's important to know that there's the gallbladder and there's bile inside, and they don't just drain bile all the freaking time. It only secretes when it's needed. And the most important hormone for this is the CCK, cholecytokinin. It controls the contraction of the gallbladder. So if you eat a lot of fatty food, this gallbladder is going to contract a lot and secrete a lot of bile. Yeah. Okay, here. Okay, so here we came to the regulation. So for the GI system, for doing all this without us paying attention to it, is because there's a lot of system to it. And if somebody asks you, it is done by hormones, a lot of hormones we've been already saying. Extrinsic innervation. What does this mean? This means, I'll explain the enteric nervous system first. So hormones And then the enteric nervous system, you can see this as like the brain in the brain in our GIT. So just think it's a mini brain. And extrinsic innervation is the one that connects this enteric nervous system to the CNS. And it is connected or communicated by the sympathetic or the parasympathetic. Yeah? Does that make sense? So we have hormones. Yeah, we have hormones everywhere. And these hormones even, I should mention that even these hormones are pretty much identical to the ones what we have in our brain. And it's actually even like we have like, for example, our happy hormone in our brain is serotonin. But somehow only 10% of the serotonin secreted in our body is in the brain. The rest is in our gut. Yeah? So yeah, interesting. That's why everybody calls it a brain, because it has serotonin, all the enkephalins, like a lot of important hormones that in our brain are in our gut. Yeah, and then we have this again, enteric nervous system. This is the brain. This is like the brain itself. It has a lot of neurons, like 100 million neurons. And it is called a brain because it can sense, and it even has a motor neuron, and it even has interneurons, interneurons. And this extrinsic innervation, which is sympathetic or parasympathetic, is the one that connects this mini brain to the brain. Okay. So that's the regulation. And now we're going to talk about the hormones that are responsible. So they love this in physiology. So we have hormones. We have a lot of hormones: endocrine, paracrine, neurocrine. And what is this? This is just a division by how it works. So this is endocrine hormone. Endocrine hormone is probably the hormone that you think of every day. So it's secreted and it works on a target cell. Yeah, it has a receptor and it works on a target cell. And it's secreted from a cell and it goes to the systemic circulation. That's what makes them endocrine. So it's all in your body circulating all the time, endocrine. Yeah, this is the endocrine fashion. And I got asked this question in physiology. I think it was during the seminar. What makes a hormone, no, what makes a target organ when we're talking about hormone? Yeah. So for example, oh, this is hard. So we have like LH, FSH receptors in our ovary, right? In our ovaries, right? So what makes them the target organ? And the answer is that if they have the receptor to it, yeah? If you don't have the receptor to it, the hormones won't know where to go or what to say to that organ, yeah? So having a receptor to it is the way of signaling it. So so not only you need hormones, the receptors are also important too. And so this paracrine one is the one that works in a local fashion way. So endocrine goes to the systemic circulation. Paracrine is only by diffusion. So it's going to secrete its hormone and it's going to work to the next cell, the neighboring cells, and all that. Okay, this neurocrine cells, you should be able to explain it. Neurocrine cells is secreted in a neuron. Oh, no. Sorry. Neurocrine hormone is made in the nerve cells, yeah, and it is transported in the axon all the way down. So it's made in the body of the nerve and it travels all the way down the axon. And only when there's an actual potential, it releases the hormone. Okay. So that's neurocrine. So you need an action potential to release that hormone. And a good example of this neurocrine is these that I wrote down, the VIP, enkephalins, and so on. This is just, I didn't write all of it because there's a lot of hormones. Okay. So Yeah, we talked about endocrine, paracrine, urocrine. So let's go to each and one of them. So gastrin, we've been talking about this. So gastrin is a hormone secreted by G cells in the antrum. And what it does is it stimulates the parietal cells to produce more H plus acid. And also, how is this G cell stimulated? It's stimulated by 3 ways. You should remember this, I think. Okay, so when there's anything in the stomach, that stimulates the G cells. Also, when there's a vagal stimuli, yeah, mediated by the gastrin-releasing peptide. I know it's a hard word, but yeah, when it's stimulated by the vagus. And also the third way is when there's amino acid in the lumen of the stomach. Yeah. Yeah, because when you have protein, it will take time to digest and you will need more acid. Yeah. So gastrin, its stimuli is mainly food, and what it does is it stimulates the parietal cells, yeah, and to produce more H plus. Okay. And in the textbook you can find this word called gastrinoma. It is a tumor that secretes this hormone gastrin. We don't want this. It's a tumor. It's a cancer. And you get too much gastrin secreted and you would have stomach problems. And this disease is called Zollinger-Ellison disease. I know it sounds very unrelated, but I remember in physiology, one of the seminar teachers taught us this, so I'm just mentioning it. So this gastrinoma, yeah? Just know that things like this could happen. You just get a tumor that just keeps on secreting gastrin, yeah? And even this tumor is sometimes not even the stomach. It could be in the pancreas, even though these G cells are mainly in the stomach. Okay. Okay. Are people okay? Okay. Just let me know if I should go slower or if you want to take notes or anything. Okay. And then the next hormone, the CCK, cholecytokinin. Yeah. It is almost pretty much the same as gastrin when it comes to the stimuli to the parietal cells, but its main action is to contract the bile duct, the gallbladder. That's why it's called cholecystokinin, like colon, as in the gallbladder, and kinin as like to move or like kinetic, that kind of word. Yeah, and also not only it stimulates the gallbladder, you want to get the pancreatic juice out to the lumen. So it also relaxes the sphincter of Oddi. Yeah, I think that's important. You should be able to say that. What else does it do? It induces the secretion of hydrogen carbonate. Yeah. Oh no, sorry. What I said at the beginning was wrong. I said it has the same like function or whatever, but I meant same structure as gastrin. Sorry, that was very different. And then so it this one stimulates for hydrogen carbonate secretion. Yeah. And also one more important thing is that it inhibits gastric emptying. Yeah. Gastric emptying. What does this mean, gastric emptying? So when you have a big meal, fatty full food, you don't want your food straight going from the stomach to the duodenum. So you want to make it slow as possible. And this, in fancy terms, is called inhibits gastric emptying. And this is what cholecystokinin does. Okay, so I'll repeat again what cholecystokinin does. So first, its main job is to squeeze that gallbladder and also open the sphincter of Oddi so that it can release the
pancreatic juice. And also what it does is this one is the one that stimulates the secretion of hydrogen carbonate in the duct cells of these organs. And also this inhibits gastric emptying, means that it just slows down everything that's happening in the stomach so that it won't go straight to the duodenum. Yeah. And CCK is, I know it's hard to remember, but it's released by eye cells. Yeah, eye cells, eye cells, eye cells. Okay, next hormone, secretin. This one I already mentioned here. Oh no, sorry. Oh no, sorry. I got confused with some other study. Okay, secretin. Secretin is it's not that easy to remember, but secretin it's the for secretion, sorry, I spelled that wrong. Secretion. It enhances the secretion of bicarbonate. Yeah. And if you think about where bicarbonate originate from, it comes from the duct cells in the pancreas and the biliary tract. So that's the secretin's job. Yeah. I know if you just read just secretin, you would think, oh, what does it secrete? Yeah, it secretes bicarbonate. Like not like a, yeah, it secretes bicarbonate. Okay, this other hormone called GIP, glucose-dependent insulinotropic peptide. Yes, it has a very fancy name, but this is just a hormone or like a hormone that is produced after eating that would stimulate for insulin production. Yeah, GIP. Yeah, yeah, so that's that. And and this one, GIP, not only it stimulates insulin production, it inhibits the secretion of hydrogen. So once again, if I repeat, gastrin and CCK increases the amount of H plus, whereas secretin and GIP decreases the amount of H plus by increasing the bicarbonate. Does that make sense? And there's this hormone called somatostatin that I already mentioned in slide 8. This one decreases the amount of gastrin, also CCK and secretin. It acts on a lot of hormones. Yeah? Okay. And also I would talk about this VIP hormone very quickly. So this is called vasoactive intestinal peptide. It's a VIP. Yeah, this one is usually in the brain also. A lot of these are also in the brain. And this one just increases the blood flow. It increases the blood flow of the intestine, the VIP. And what it does and by that it relaxes the smooth muscle in the GIT. Yeah, that's VIP, vasoactive intestinal peptide. Okay, I know it's a lot, so I put this slide so we could revise. So this one we're talking about the gastric phase, and then here, so the next one, it's the intestinal phase, yeah? And you would know why it's different, why it's separated, because you could see here that it has different things. So when you eat, there's food in the stomach. That is immediately stimulates gastrin secretion. And by that, it increases motility of the GIT and increases acid secretion. Yeah, we did this, gastrin, more hydrogen acid. And then because of this, then it will go, the food and the acid will go to the duodenum. Now you want to neutralize this acid. By how? By secreting CCK and also also secretin and GIP. Yeah, this secretin, GIP, all responsible for lowering the lowering the acidic level. Yeah, so CCK, secretin, and also what it does is also, as I said, pancreatic and biliary secretion, which causes intestinal digestion of the food. Yeah, and these are pictures from Ganon, and I did use a lot of Ganon, and also for my actual exam I used Ganon, and the lectures. Yeah, I know a lot of people do use big books, but for me it was enough because I don't like putting too much information and making myself even more confused. So yeah, these pictures, and it's a nice way of revising. Just see the picture and be like, can I explain what is going on here? I think that's how you should study physiology, or that's how I studied it, because you should be able to tell the story from the top to the end. You should be like, so this happens, and then this happens, and that's why this happens, and at the end you get this kind of thing. Yeah. And I also attached this picture to know which hormone is secreted where. And this is important because when I was studying this, I thought all these hormones were secreted in the stomach. But actually, it's not. Only gastrin is secreted in the stomach. Everything else is below the stomach, yeah? All the CCK secreting, GIP, motility, all the other ones, they're secreted all below. And gastrin is an exception. It's secreted by the antrum. Yeah, and this should be the one that's working first. That's why. Yeah. So I just wanted to attach this to let you know that these hormones are not secreted in the stomach, but all the intestinal cells below that, like in the duodenum, jejunum, ileum. Okay. Next one. So I already said a little about the enteric nervous system. So I should talk more about it. So, enteric nervous system. So this is like the other name of the second brain. It's called a brain. And after you die, or even a dead animal, if it just died now, if you open up the intestine, that it's still moving. And why? Because it doesn't, like the basic function of the intestine doesn't require our brain. It's just a local reflex. It's going to do its own job on its own. Because it has, what does it have? On its own, it has sensory neurons, interneurons, motor neurons. What does that mean? It means that sensory neurons, it senses that food is here. It senses that, oh, it stretched this bit here, so I should relax now so that the food goes forward. Yeah. So it senses and then it moves accordingly. So that's why it has a motor neuron. Yeah, and that is exactly what our brain does. You know, you touch something hot and you try to avoid it by the motor neuron. So this is why one of the reasons why it's called the brain. And also, as I said before, this enteric nervous system is not entirely independent. It is connected with the brain. How? By the sympathetic and the parasympathetic. So, for example, before the exam, yeah, you have crazy gut movements, GIT movements. Why? Because you're in a sympathetic mood, and your GIT is influenced by that. Yeah. So this connection between the brain and the gut is called the gut-brain axis, and this like stimuli from the brain is called extrinsic because it's outside of the brain. And all happening inside the brain without any external stimuli is called the intrinsic part of this enteric nervous system. Does that make sense? So this enteric nervous system, you can further divide into two, extrinsic and intrinsic. And and extrinsic is the one that connects with the brain, and intrinsic is just the local reflex. And this is important. So it's by the sympathetic and the parasympathetic, we know that. And the sympathetic is usually in our body, it's excitatory. Every other organ it's excitatory when it's sympathetic. Your heart, it starts beating fast. Yeah. But in our gut, it's the opposite. It's, if you think about it, yeah, everything that doesn't have to do with like fighting, running, that kind of thing is relaxed more, and when it's sympathetic. Yeah, so yeah, sympathetic. So it's, you could say sympathetic is inhibitory and parasympathetic is excitatory when it comes to the GIT system. Or you could talk about the glands as well. Yeah, it's the same thing. And where does the sympathetic nerve come from? And it is by T8 to L2. So you know there's the sympathetic ganglions next to the vertebra, and which level? Oh, sorry. It was like, which level? It's from T8 to L2. Yeah. And so it goes to the prevertebral ganglia and goes all the way to the submucosal and the mesenteric plexus and affects secretion and motility. And parasympathetic, on the other hand, is excitatory, and it is done by vagus and this pelvic plexus. As I said before, vagus is all the way down to the upper large intestine, and from down there, so the lower, so for the descending, large descending colon, sigmoid colon, rectum, anus is done by the pelvic plexus. So that is important, this level, what kind of effect it has on the GIT. So sympathetic and parasympathetic. Yeah. And then this intrinsic one is the one that it keeps on moving even after you die. And yeah, it's a local reflex. So by this two, it does its own job. It does its own, yeah. Sorry, wait. And this is the same thing that I already said. Submucosal plexus, it's in the mucosa. What's in the mucosa? Glands. So it controls intestinal secretion. Whereas the mesenteric plexus is in the muscle. So what does muscle do? They contract and make either mix the food or the bolus that's already there, or make it go forward. So it controls the movement. And food, when you eat, when it's still like a thing inside the esophagus, it's called a bolus. And when there's a digestive juice mixed and already liquidish, then the name changes to chyme. So I think you would read that a lot in the books. Yeah. So bolus and chyme, they both mean like food that's already in the system. That's already in the GI system. Okay, so let's talk about peristalsis. So this is the main thing that's happening in the GI so that the food goes forward. And when I thought peristalsis was just this, if you can see what I'm pointing at. One part contracts and the food is here, and then the one forward relaxes, and that keeps on going the food to go forward. Yes, that is also peristalsis, but there's this different type of peristalsis called segmentation. Yeah, segmentation. And this is very important for mixing. Yeah, you need to mix food with whatever is secreted already to help digest, not only in the stomach but also in the intestine, so that you absorb better and all that. And segmentation happens when two parts of the circular rings contract, so like this, here and here. And then when there's like this bit formed, a room formed, it just contracts in the middle. And what happens is that the food bounces, and that makes the food mix. Yeah, so this is mixing. segmentation, peristalsis. Yeah. And what else should I say? You can probably imagine this contraction is done by the circular muscle, and this whole movement is done by the longitudinal muscle. So imagine all the longitudinal in the intestine shortens or contracted. It makes the GI very short. And if it relaxes, vice versa. Yeah. And whereas the circular muscle, it just contracts at that point so that it pushes the food forward. Yeah. And this is a good example that I wrote. So yes, it can do all this on its own by its local reflex, but when does the sympathetic and parasympathetic does its job? So if it's normal, you just enhances or inhibits in a regular form. But when these goes crazy, so before an exam, before fighting, during war, whatever you can imagine, it leads to crazy things as like vomiting and diarrhea. Yeah. And this is when you know that there's a connection between the brain and the GIT, yeah, by the sympathetic and the parasympathetic. Yeah, and I talked about the segmentation. And yes, this is I think I already said it. So local stretch releases serotonin. I think the stretch receptors gets activated. It secretes serotonin, then which activates the sensory neuron. And that activates the mesenteric plexus, which is motor neuron. So yeah, that's how we digest, or that's how peristalsis happens. And you probably be wondering, like, how does it do it on its own? And of course, it's physiology, so they're gonna want to know that. So if you think about the heart, it's the same thing. It's gonna keep on beeping on its own without us controlling it. And why is that possible? It's because we have a SA node, and it generates electricity on its own, and all the heart cells, myocytes, are electrosensitive or electroconductive. So that's how the heart's beating. With the GI, it's similar, but it's a little different. As in, I want to look for my notes. Well, maybe I'm okay without it. So we have pacemaker cells in the intestine, same as in the SA node, called the interstitial cells or intestinal cells of Cajal. Yeah, this is one thing you should remember. Yeah. And this pacemaker cells set up this baseline rhythm that you could see here. I don't know if you can see properly, but this is the oscillating slow wave pattern. Yeah, this is all because of the intestinal cells of Cajal. Yeah, and usually when you're resting, normally the resting membrane potential is -50. Yeah, this is where it is. And when you're eating, or when you have parasympathetic stretch, acetylcholine, these kind of stimuli, it's going to bring these resting memory potential a little higher up to here. Yeah. But still, for us to have that contraction, of the intestine, we need these active potentials, these spikes that you can see. Yeah, these spikes. These spikes. And these spikes cause, if they reach a certain amount, it makes these cells depolarize and you get the thing you want. Yeah. But when you don't want, so when you're in a very sympathetic state, you're here. So you're going to go down, hyperpolarize. So it's going to be harder to actually make one actual potential all up to here. It's going to make it hard. You could still get the spikes here, but it won't reach all the way up here to 0 or -10, and it won't depolarize. Does that make sense? I think I can explain better, but I think I feel like I'm doing that. Okay. So there's this baseline rhythm. This is slow wave that is resting around -50. Yeah, and this is all because of intestinal cells of Cajal. And when we have these stimuli from the mesenteric plexus, or depends on the stimuli, it could be stretch, acetylcholine, and parasympathetic. First of all, the baseline is higher. And when you get the spikes, these spikes, it's so much easier to depolarize and get to the threshold. Whereas when you have the opposite scenario, when you have a lot of adrenaline and you're in the sympathetic state, this slow wave, the baseline just goes down, so it's harder for you to reach up here. Yeah. So this is the explanation for this this picture. And also for contraction, you think it was just this, but actually there's two kinds of contraction. This is important. So there's tonic contraction and phasic contraction. The difference is tonic contraction, it's contracted all the time. Whereas phasic contraction, it contracts, release, contract, release, contract, release. And why? This is more for mixing. Yeah, this is more for mixing. So it happens in the intestine, or sorry, in the stomach. Whereas this tonic contraction is more for the sphincters. Yeah, sphincters. Or like, where else? Sorry, I've forgotten, but I think you should look up if you have your book. And there's a picture on which part of your body does tonic contraction and which does the phasic contraction. But how I remember is that one is for mixing, this one is for mixing, and the tonic contraction is more for keeping that tension. Yeah. Oh, I'm glad I wrote it down here. So, sorry I forgot to mention, going back to the slow waves. The slow waves by the Cajal, it goes up by the sodium influx and goes back down from potassium efflux. Yeah, I think that makes sense. But when it comes to the spikes, it's from calcium influx here. And and when it goes down, it's the same as this going down. So remember, these spikes are done by calcium influx. That is important. So it's different. The mechanism is different. This one is sodium, potassium, but this one's calcium and potassium. Yeah. And what I wrote here I already said. So what stimulates the action potential? So parasympathetic tone, vagus nerve releasing acetylcholine, and stretch itself is also a stimuli to activate serotonin and actually causes these local reflexes. So that's this. Okay. And this one, migrating motor complex. This is a word that you should remember. And what it is it's basically when you're hungry or when you haven't eaten in a while, your gut just has its own peristaltic movement from the stomach all the way to the jejunum, or maybe to the colon as well. And what it does is that it swipes away all the residual food that was there. So for example, you eat, I don't think you eat bone itself, but imagine if you accidentally ate bone or a lot of fiber. They get stuck, or they take so much time to go through the intestine. But by this migrating motor complex, it can go forward. And this happens from the antrum. This antrum starts this whole thing, this electric activity, and contracts. And then from the antrum, it goes all the way down to the jejunum. Yeah, I think. And then you should know what these waves are made from. So, when it's stimulated, it's phase 3. Can you see this? So when there's increased electric activity, that you can see these spikes, and it causes contractions. And then afterwards, it's phase 2, when there's like little spikes, like probably here. And then when it's just in the baseline, it's phase 1. Yeah, so basic electric rhythm is there, but no contraction is happening. So that's this, that's this, that's this, that's this. And yeah, so this one, yeah. And also, bear in mind that it happens after you eat. No, like, yeah, it happens a while after you eat. So it happens when you haven't eaten a while. So, and it happens if you haven't eaten, it would happen every 2 hours or something. It said 90 to 200, every 240 minutes. So, yeah, like, I would just remember it happens every 2 hours, and it would stop if you eat, obviously, because this one happens when you're hungry and when you're in a fasting state. Oh, and yeah, remember this is stimulated by this hormone called motility. Motility. It's a hormone, and its job is to do this motility, and it has motility. It's in the name too, so I think it's not that hard to remember. Okay, so next to the swallowing reflex. Okay, swallowing reflex, if you get this question in the exam, you should be able to say what happens from the beginning to the end, like as a story. And what's important here is that when you swallow, you don't really think, but it's a lot of things happening at once, and a lot of muscles are coordinated. Yeah. And how is it coordinated? It is coordinated by nerves that are innervating the muscles, and it all comes from this swallowing center called in the medulla oblongata. Yeah. And you gotta think that the trachea is right in front of the esophagus. So you don't want food in your trachea. So you're gonna make sure that you will be breathing as normal when you're eating, but you're gonna stop a little bit and cover the epiglottis and close that opening. And also when you swallow, you don't want that food to be going somewhere like in the nose or pushed back in front of the mouth. Yeah, so we'll talk about what happens. So when we're swallowing, it happens from the medulla. Yeah, remember this. A lot of basic things of like reflexes are done in the medulla oblongata. Yeah, and so if you remember from histology or in anatomy, a lot in the mouth, it's in the mouth, your voluntary muscles move, so your tongue is voluntary, and esophagus is mixed, as in like upper 1/3 is striated and the lower is smooth muscle, which means smooth. So that's also important to remember. And depending on where the food is, we can divide this full swallowing reflex into three, which is the oral phase, pharyngeal phase, and the esophageal phase. Depends on where the food is, yeah. And oral phase, we think about this. We swallow with our own decision. So what happens is that we use our tongue to push the food backward, and we keep it there. So we keep the tongue there so that the food doesn't come back to the oral cavity. And we also close or the soft palate also pushes down so it doesn't go back to the nasal cavity, or it doesn't go to the nasal cavity, sorry. And in the pharyngeal phase, this is where the tactile receptors are. Tactile means like it has receptors that you know that you touched. And they know that the food is there, and what it does is that it covers the epiglottis. It covers the trachea with the epiglottis. Yeah, so this is all reflex. So the sensory is from the tactile receptors sensing the food existence, and then by reflex, the epiglottis covers the trachea. Yeah, and then the food can go down. And food goes down by peristalsis. So the muscle behind the bolus would contract, and muscles that's in front of the bolus would relax. That's peristalsis. And on top of peristalsis, we have gravity that pushes the food downwards. But what they like to say is that, oh, even in space, you could still swallow because it's not just gravity that's working, it's also done by peristalsis. Yeah, so that's where it comes from. And also the esophagus, the esophageal phase has two parts. So when you swallow, it goes down and it opens the lower esophageal sphincter that connects to the stomach. Yeah, it relaxes so that the food goes in. That's the primary one. When you just swallow normally, it takes a couple of seconds and it goes down. But sometimes there's a residual that's left in part of the esophagus. We don't want that. So there's secondary reflex that just pushes whatever that's left down to the stomach. So you should know that there's primary and secondary esophageal reflex. Yeah, yeah. And also for the lower esophageal sphincter, it's usually always closed only when the food is right in front, right above it. And it has two sphincters, internal and external. And internal is part of the esophagus, but the external sphincter is part of the diaphragm. So if you could check that when you're studying, that would be nice. And the difficult part here comes here. So I just talked all this without mentioning which nerve censored it and which nerve actually worked on which muscle. So you could use this picture. Yeah, so when it's in the oral cavity, you sense it by the 5 trigeminal nerve, the mandibular branch, because it's here in the mouth, and also in the glossopharyngeal and the vagus. Yeah, and then it goes back to the medulla oblongata, and then as a reflex, it sends signal on 9, 10, and 12. And if you think about the muscles here, you would understand. Yeah. So if I get this on the exam, I would remember which muscle was which, and you talk all about what happens. Sorry, when you after you eat, what happens to the palate, what happens to the epiglottis, what happens to the laryngeal muscles, and you should know which nerve innervates which. Yeah. Was that helpful? I hope it was. Yeah. So just know there's sensory and there's motor. And it's a reflex, so it's done by the medulla oblongata. It goes in and out. And which muscle does it innervate? Yeah. And this peristalsis movement is important of the esophagus, and also gravity plays an important role. Yeah. Okay, I'll go to the next one. So vomiting. I won't go in much in details, but for vomiting, there's a lot of trigger. So when do you vomit? So sometimes you vomit people that are pregnant, or also when you get classic motion sickness, that comes from the CNS. But if you put your toothbrush up to your throat, that also causes vomiting. Why? Because it has mechanic receptors that tells the vomiting center in our brain to vomit. So as I wrote here, trigger could be a lot of things. So limbic system, mechanical irritation. And this vestibular nuclei is the one responsible for motion sickness, because you get motion sickness from your vestibular system in your ear, and that stimulates the vestibular nuclei, and you have the sensation of vomiting. And also chemoreceptor, this is when you get intoxicated or things like that. You feel like vomiting because of the chemical property of the stomach, or in the blood too. So there's different kind of triggers. So either, yeah, it's CNS itself, or it could be from somewhere else, but it all goes to the vomiting center in the medulla. This again is in the medulla oblongata. Yeah. And what happens next is, well, you have the sensation of nausea, and also you salivate a lot. Yeah. And you should know that vomiting starts from the intestines. Not in the colon, not in the stomach, it starts from the intestine. And we learnt swallowing, yeah? And if you know the swallowing mechanism pretty well, you just have to go backwards. Everything backwards is vomiting. So you just start from the jejunum, the duodenum, and then there's a pylorus, so that has to open. And then it goes, and the antrum has to relax so that it comes out. And also you have to increase your abdominal pressure, and you do that by contracting the diaphragm. And yeah, so you just go backwards. So you relax the cardia so that the food gets in from the stomach to the esophagus. And and at the end, you have to relax the upper pharyngeal sphincter. Oh, I forgot to mention that esophagus has two sphincters, upper and lower. I think you know, it's all right. Okay, so vomiting, what's important is that it starts from the intestines, yeah? So you stimulate however you want, and it goes whatever the way, it goes to a vomiting center in the medulla. And as a response, it starts from the jejunum or the duodenum, and it goes backwards. Yeah, everything you learned at swallowing, it just goes backwards. Yeah, and I remember our teacher explaining it that way, so I remembered it that way too. Okay, colon, I won't go much in detail. So colon, what its main job, they have a lot of bacteria, by the way, yeah. And so we don't want the colon specific bacteria to be in the intestine. And also, yeah, so that's why there's the ileocecal valve. This is the one that's connecting the ileus to the cecum. Yeah, so it means that it's not just flowing all the time. It only opens sometimes for the content to go to the colon. Yeah, so that's ileocecal valve. And it's usually closed. That's what I wanted to say. And its main job is to absorb water. 90% of the water is absorbed in the colon. And like we said, in the peristalsis, we covered segmentation and the peristalsis. But other than these two, it has this mass action contraction, which is a colon specific. Yeah. What I imagine imagine this is like a massive peristalsis that happens only in the colon. Yeah, after you drink coffee, I think this is what it's happening. Yeah, and this is what I mean, gastroileal reflux. This is when you stimulate the peristalsis of the colon or the ileum from the from the stomach. And this is done by the vagal nerve. So any vagal nerve reflex is called a vasovagal reflex. And same thing, after you eat, there's the I forgot, but also the stomach can influence the peristalsis in the rectum or the descending colon. And that is also a vasovagal reflex. And that is why